Hi and welcome to this revision video on the potometer practical. A potometer is used to measure the rate of transpiration in a plant. Transpiration is the loss of water from the leaves. As the leaves lose water, the plant will replace that water with water from our reservoir, pulling it through the capillary tube. And we're going to change the environmental conditions to see how this affects the rate of transpiration. In transpiration, water will move from the roots, which is simulated by this dish of water, through the xylem, which is simulated by the capillary tube, through to the leaves, where it will be lost via evaporation through the stomata. And as it's lost through the leaves, it will replace that lost water with water from the stem, which is then replaced with water from the roots. And what we can see in the capillary tube is a tiny air bubble which will move along the ruler, and we can measure both a start and end point and calculate a rate of transpiration.
First of all, we're going to run the experiment with our control conditions before repeating with increased light and increased airflow over the plant. Before starting the timer, we have to record the starting position of the air bubble. You can see here that the bubble started at 49mm along the ruler. It would be ideal to start with the bubble at 0, but this isn't always possible, so just recording the start and end measurements and calculating the difference is the next best thing. Now let's start the timer and see how far the bubble moves in 5 minutes. Don't worry, it was recorded as a time lapse, so you won't be here too long. The experiment was stopped after 5 minutes and the final position of the bubble was recorded. You can see here that the bubble reached 81mm along the ruler. Note that the position is measured from the same point on the bubble each time to ensure that the results are valid, and the smaller the bubble, the better. We can use the start and end points to calculate the distance travelled by the bubble over the 5-minute period. The start point was 49mm and the end point was 81mm. To find the total distance, you need to subtract the start point from the end point. So for this one, that is 81 minus 49, giving a total distance moved of 32mm. This was the control experiment, which was done so that there can be a comparison made after the conditions are changed. We can use this information and the data we gather in the next 2 experiments to calculate the rate of transpiration. We'll do that later and compare all the rates at the same time.
I'm now going to change the environmental conditions on the plant using this lamp. The increased light intensity will increase the rate of photosynthesis, therefore more water should be taken up by the plant.
Now we are going to look at the rate of transpiration when the light intensity is increased, which would represent a sunny day for the plant. This time the air bubble started 72mm along the ruler. Let's take a look at the time lapse for this one and see how far the bubble moves this time. And then we can use this to calculate how far the bubble travelled when the light intensity increased. After 5 minutes, the bubble was at 109mm along the ruler. Again, let's calculate the total distance travelled by the bubble. The start point this time was 72mm and the end point was 109mm. Again, to find the total distance, you need to subtract the start point from the end point. So for this one, that is 109 minus 72, giving us a total distance moved of 37mm. So a slightly greater distance this time. We'll record this value and use it later to calculate the rate of transpiration.
I'm now going to change the environmental conditions a second time using this hairdryer. The increased airflow will mean that the water that leaves the plant through transpiration will be removed, and this will maintain a steep concentration gradient between the inside of the leaf and the environment.
Final experiment, we used a hairdryer to increase airflow around the plant. This would be like a windy day for the plant. This time, the starting point of the bubble was 47mm along the ruler. Let's look at our final time lapse to see what happened. At the end of our final experiment, the bubble is at 102 millimetres along the ruler. As with the previous experiments, we need to calculate the total distance travelled. The start point this time was 47 millimetres and the end point was 102 millimetres. Again, to find the total distance, you need to subtract the start point from the end point. So for this one, that is 102 minus 47, given a total distance moved of 55 millimetres, so quite a bit further than the previous two experiments. Now we can use the data we collected to calculate the rate of transpiration for the different conditions. To calculate the rate of transpiration, the equation used is distance travelled by the bubble divided by time. For this experiment, we're going to give our answer in millimetres per second, but it may vary depending on the question. Make sure you pay attention to the units asked for. We can use the distance travelled by the bubble as a measurement of the rate of transpiration because it's shown us how much water is being drawn up by the plant's roots as water is lost from the leaves through evaporation. The time for all of our experiments was the same at 5 minutes, but we need to convert this to seconds by multiplying by 60, giving us a time of 300 seconds for all experiments. To calculate the rate for the control experiment, we need to do 32 divided by 300, which gives us a rate of 0.1 millimetres per second. For the lamp, we need to do 37 divided by 300, giving us a rate of 0.12 millimetres per second, which is slightly higher. And for the hairdryer, we do 55 divided by 300, giving a rate of 0.18 millimetres per second. So that gives us the highest rate of transpiration for the hairdryer. Now it's time to take a look at some exam questions on this topic. So question 1. Some students use the apparatus shown in the diagram to measure the rate of water uptake by a plant cutting. And you can see there a diagram that shows the equipment that we were just using. It's very, very similar. They can show it in a couple of different ways, and I think the next example has a slightly different diagram on it again, but they all go with the same idea of having some sort of water reservoir, an air bubble, a ruler, and of course this capillary tube with the plant cutting at the end of it. So the students set up the apparatus in 3 different conditions: no wind at 15 degrees C, no wind at 25 degrees C, and wind at 25 degrees C. So for each experiment, the students recorded the movement of the air bubble along the scale. Name the 2 variables the students chose to change in these experiments. So of course, the variable that you change is your independent variable, and there are 2 independent variables for this experiment. So we have the temperature, as they looked at results at both 15 degrees C and 25 degrees C, and also wind. So whether there was wind or not for the different experiments. So they were the 2 independent variables. It was important to use the same plant cutting each time to make these experiments fair. Explain why. So now this question is asking us about one of the control variables for this experiment, and probably the most important control variable. So the plant needs to be the same because you could have different sized plants, you could have plants with a different number of leaves, and all of this is going to lead to plants that have a different rate of uptake of water. So something along those lines. So you could have said that different plants have different sizes, different number of leaves, or different uptake of water, which would affect your experiment, and it would mean that you wouldn't be able to accurately see what the effect of the different independent variables was. So you need to use the same plant. The graph here shows the students' results. So which line on the graph A, B or C shows the results for each of the 3 different experiments? So we can see here that line A shows that the air bubble travelled the largest distance. So for line A, the transpiration rate would have been the highest. Line B was the second highest rate of transpiration, so it travelled about 46 millimetres in the same time that for line A travelled 68 millimetres, and then line C, it travelled about 23 millimetres in the same amount of time. So that would have been our lowest rate of transpiration. So we need to write the correct letters in the correct boxes. So no wind at 15 degrees C, so we have the lowest temperature and also that there's no wind. So this one would be line C. So this is going to be our lowest rate of transpiration, which is kind of similar to what we saw in our own experiment where when there was no wind, it was the lowest rate of transpiration. No wind at 25 degrees C is probably going to be a little bit higher. So if it's warmer, you're going to get a little bit more transpiration going on. You're going to get a little bit more evaporation of water from the leaves. So that would be B. And wind at 25 degrees C is going to be A. So you're going to have lots of evaporation going on when it is at 25 degrees C because it's going to be warmer, and also the wind is taking away that water vapour and maintaining your concentration gradient so that more water can diffuse out of the stomata and keep that process going. So A is going to be our greatest rate of transpiration. Water is lost from the leaves of the plant cutting. Name this process. Draw a ring around one answer. I'm going to delete the other two. It is, of course, transpiration that results in the loss of water from the leaves. So let's move on to question number 2. A potometer is a piece of apparatus that can be used to measure water uptake by a leafy shoot. Figure 1 shows a potometer. So this is an even simpler example. So it's purely just a capillary tube with a scale on it, connected to this tubing with the leafy shoot in the other end of it, all attached to a clamp stand. It looks different. It works in the exact same way as the one that we've looked at and the one in question 1 as well. So some students used a potometer like the one shown in figure 1. They measured the water taken up by a chute in normal conditions in a classroom. As the water was taken up by the chute, the level of the water in the capillary tube went down. The students recorded the level of water in the capillary tube at 2-minute intervals for 10 minutes. So here we go, we've got our time and our water level in millimetres, and it tells us the area of the cross section of the capillary tube was 0.8 millimetres squared. Okay, so if you wanted to find the volume, which is what it's going to ask us later on, we need to be using this area of the cross section to be able to give our measurements in millimetres cubed. So complete the following calculation to find the volume of water taken up by the chute in millimetres cubed per minute. Now we just gave our rate of transpiration as how many millimetres did the water move in a given time, but we could convert that to a volume, and they may ask you to do that as they have in this question. So first of all, distance water moved along the scale in 10 minutes. So it didn't start at 0, so this is really important. Our scale did not start at 0, much like our own didn't. So we need to find the difference between the start position and the end position. Now if you remember when we looked at it earlier, we do the end position, subtract the start position, which would give us 5 millimetres. So next, volume of water taken up by the chute in 10 minutes. So this is where the area comes in, okay? To find the volume for the water that was taken up, we need to multiply the area by the length, because we're kind of thinking of it as like a cylinder, a 3D shape. So we're going to do 5, I'm going to type this out, 5 times 0.8, which would give us 4. Okay, so that is 4 millimetres cubed, and that is over 10 minutes. Therefore, the volume of water taken up by the chute in 1 minute. So we need to divide this by 10, of course, because we only want 1 minute instead of 10 minutes. So we're going to do 4 divided by 10, which gives us 0.4 millimetres cubed. So that's the volume taken up per minute. So our final question, the students repeated the investigation, but this time placed the potometer next to a fan blowing air over the leafy shoot. Suggest how the results would be different, give a reason for your answer. So this is very much like when we use the hairdryer to change the conditions for our plant in our own experiment. And what we would see is that there would be an increased uptake. And the reason why for this would be because there's a greater rate of transpiration, because there is more evaporation of water from the leaves in those conditions. So it's only a 2 marker, so it only needs to be a short description. So increased uptake for our first mark, because there would be increased evaporation. Thank you for watching this video. That is the end of the exam questions, and I hope that this was a helpful look at the potometer.